one of the refresher course in life science organized by UGC HRDC Goa University. This afternoon, introducing our resource person is my great privilege. We have amongst us the eminent personality, Professor Ashwani Parikh. Professor Ashwani Parikh is Executive Director of NABI, that is National Agri-Food Biotechnology Institute, a prominent plant biologist and educator noted chiefly for his contribution in plant molecular biology and biotechnology. Sir is currently working as Professor of Plant Molecular Biology and Biotechnology at the School of Life Sciences, JNU, New Delhi, and an adjunct professor at the University of Western Australia, Perth, Australia. He is a recipient of several honors, including the Visitors Award for Technology Development from the President of India for developing stress-tolerant rice of the next generation that is strong. His research areas of interest and specialization include functional genomics, forward and reverse genetics, abiotic stress, and crop biotechnology. Sir has received various awards and honors. To name a few include National Academy of Science, Reliance Platinum Jubilee Award in 2016 for industry-oriented research in biological sciences, J.J. Shenoy Gold Medal Award in 2016 by Indian Society of Plant Physiology for contribution in the area of plant science, Fellowship Award in 2017 from BioVed Research Institute of Agriculture and Technology, Allahabad. Sir is the editor in charge of for Indian Journal of Plant Physiology, Springer. Sir was elected Fellow of the National Academy of Agricultural Sciences, New Delhi in 2016. Sir has 11 patents to his credit. To name a few recent ones include hybrid type histidine kinase gene isolated from indica rice and clones produced therapy that was granted by Indian grants in 2016. A method for cloning a hybrid type histidine kinase gene and an expression vector compromising the hybrid type histidine kinase gene in 2018 from Vietnam.
So, Desilia, I think
So,
that's so much is not required.
okay, so in short,
Thank you so
just
much.
yes,
I'm so humbled to be present among the extreme group. So we can move on with the next agenda. Thank you very much.
yeah, welcome you, sir, on behalf of all of us.
Thank you, Desilia. Sandeep, do you think I can start now?
Yes, sir.
Yes, yes, please.
One of you need to tell me if my slides are visible and I'm audible enough.
Yes, sir.
Okay, I'm trying to share my presentation. Are you able to see the full slide now?
Yes, sir.
Okay,
Yes.
wonderful. Thank you very much. Very good afternoon to all of you. I think what all has been said by Desilia is something which is not true, I would say. The truth is that we are here together and trying to learn something which is going to be a very wonderful exercise. My name is Ashwani Parikh, and currently I'm holding several caps, I would say, at the Department of Biotechnology, Jawaharlal Nehru University, and a couple of other things. So I really want that let this lecture be a sort of interactive session. Wherever you think I'm not clear enough, I would expect that you will be stopping me and asking a question in between as well. But since I said I have to go to the airport around 3:30, I would really be happy if you can pen down your questions, and we will have enough time after 3 o'clock to answer some of your queries. So I'm not going to talk about the research which is being done at NABI, which is National Agri-Food Biotechnology Institute under the Department of Biotechnology. Another institute is CIAB, Center for Innovative and Applied Bioprocessing, or any other place which I'm showing over here. I'm going to talk to you about something very, very general, something which is concerning each one of us and every one of us who's listening to me. I'm told this is a refresher course which is being organized by the Goa University, and I know that all of you belong to different disciplines: physics, chemistry, biology, biotechnology, microbiology, agriculture. So I'm trying to be as general as possible, and in this process, some of the information which you will find will be very, very basic, very preliminary, but bear with me if you find something very, very basic. It is just to, make the background available to all those who are not from the field. Having said that, I'm going to talk to you about a very important title which talks about, are we ready to feed 9 billion? And I have very specifically and purposefully picked up this title, and which will be very, very evident and clear as we move along this lecture. I would like to start my lecture by giving you something very, very fundamental, which I picked up from public resources. This is talking about the capacity of human brain in terms of doing observation, in terms of asking questions, in terms of developing technology, in terms of finding solutions to the problem. Yes, I'm talking about Homo sapiens. Obviously, we have all wonderful brain, and we all belong to it. And I believe that we almost evolved 300,000 years ago. And since the evolution of human brain, we are expanding our brain and brain activities, asking questions, finding solution to that, and we are actually changing the environment around us, be it in terms of physical environment, chemical, geological, and most importantly, the biological environment we are talking about. When we are talking about these things, that when we are trying to make our life more comfortable, we are doing the Herculean task, as I'm showing in the next slide, which I picked up again from the public resources. This is a very interesting slide which talks about there is a mountain. If you see the image on the left side, the image was taken in the year 1984. If you can see, the boundaries shown over here show the big mountain, which we call this is Hobart Coal Mine Mountain over there, which is in West Virginia. And because of the excavation activities, not only the whole mountain has been removed, but there is a big there is a big cavity over there, which is around 10,000 acres, which is shown in the picture on the right side in the year 2009. Why am I showing this picture? Because I just want to share with you that, we could do this Herculean task because we thought of it, we took it as a challenge, we used the available tools and technology, and then we developed a solution, and finally we could achieve the target. The importance of tools and technology is the one which I'm going to stress about. When it comes to handling any kind of issue, be it in terms of removal of mountain. Shown over here is another image. This talks about Three Georges Dam, which is the world's largest hydroelectric power generator. The reservoir is actually more than 2 miles, 3 kilometers across. And if you see the picture on the top left side, which is showing the picture of the river in the year 1987, the river is moving very smoothly with almost uniform pattern on both the side. But because of the creation of the dam in the year 2006, if you see, of course, there is the expansion of water table, but if you see the erosion around, and of course, as a student of biology, I agree that, and I believe that a lot of biodiversity has also been distorted or affected, at least I can say, by the formation of this. But nonetheless, the point I'm trying to make over here is that we could remove the mountain, we could control the mountain, we could control the rivers, and we could actually do this because we have the tools and technologies available when it comes to handling the Herculean task. Some of these tasks are not so good in the sense that because of the human activities, we have lost a lot of birds and animals. Shown over here is an example of dodo's extinction, which is Raphus cucullatus, and it got extinct almost in 17th century. The beautiful bird is shown on the right side. And some of these activities of human brain are actually very extraordinary, I would like to say. And shown over here is an example of creation of human mind, which we are creating the living beings, which are really, really extraordinary. And sometimes we need to ask the question that why we are doing it, are we doing it the right way, and so on and so forth. But nonetheless, the point I'm trying to make over here is that there are tools and technologies available almost to handle any kind of challenge you face in the years to come. Shown over here is coming closer to the topic of today's discussion. I'm going to show over here is an image, and some of you will agree that it looks like a bird, white dove, and some of you will, those who are from botany, they would agree that this is not a bird, this is actually a flower coming from a plant. But nonetheless, as a molecular biologist or as a biologist, we all agree that when it comes to the basics of life, we are all equal in terms of DNA, RNA, and protein. Be it microbes, be it plant, be it animal, be it birds. We have the DNA, which is comprising of 4 base pair: adenine, guanine, cytosine, thymine. We have learned it in the school level. When it comes to the making copies of DNA, we do transcription, and RNA is coming out where thymine is replaced by uracil. And when it comes to making the protein, this messenger RNA in the transcript is actually translated by ribosome, and what we get is the protein. Proteins are the one which determine the homeostasis in the cell, that determines the physiology, and finally the living being, be it in terms of birds, animals, or human being, or the plant we're talking about in this particular context. But nonetheless, the science has given a confidence that we have understood the basics of life, and we are confident that we can not only read it, we can not only understand it, but we can use it for the benefit of mankind, where we can manipulate one or two bases or the pieces of DNA and get the desired result, which otherwise would have been considered an Herculean task had we not knowing the enzymes, the molecular biology, the principles of central dogma of life behind it. Having said that, coming to the today's topic, which talks about the plant. Plants are very dear to me, at least I can tell you, because I deal with plants in the morning. I get up in the morning and I go to bed in the evening, because my food, my fiber, which I use it at the cloth, my drugs, pharma, the fuel, everything actually is coming from plant. The plants are very, very important for us, and that is where we need to understand that since we have good amount of supply coming from the plant, which is in the form of food and fiber, can we ensure these food and fiber coming in 50 years from now? And that is the question, the title which I said. Are we ready to feed 9 billion? Because we're going to be 9 or 10 billion very, very soon, as far as this global green earth is concerned. When it comes to handling such questions, which are again Herculean in terms of biology, making food available for 9 billion, we have to understand the history behind it. Why are we able to ask this question? Have we done it in the past? Do we have enough technologies available, which has given us a confidence that yes, we can still go ahead with it and continue harvesting the benefit of plant in terms of feeding the 9 billion? Here comes a technique which is shown in the image. On the right side, you have a flower coming in, which is emasculated. On the right side, on the left side, you have another plant of brassica, which is being used for the pollination. This process, where you can bring in the pollen from a particular plant, particular variety, and putting on to the female part of any other plant, is actually a kind of control with production. And the biologists prefer to call it plant breeding, because they are able to breed a plant, like animal breeding, where you can actually make the cow and the other part of it, or buffaloes around it. You can actually do plant breeding as well by understanding what time the pollens are coming out, by what time the stigma, the female part, is receptive, and you can actually do it in a very, very beautiful way and get the desired product. Since I assume that you understand the basic biology, this plant breeding has given us enough confidence. The new varieties of crop, be it in terms of rice, wheat, tomato, maize, whatever you're talking about, this is actually an outcome of simple understanding of pollen and the stigma biology. But nonetheless, we also understand that every technology has a limitation, and the limitation over here is that we can only cross the plant which are sexually compatible. What I mean by sexually compatible is that where the pollen of particular plant or variety is able to go to the stigma and fuse with the egg cell in terms of the nucleus we're talking about. Sometimes, if you go beyond the sexual compatible plant, you are not able to achieve the desired product. For example, if you want to cross mango with rice, you cannot do that. And within rice, if you see, all the varieties are not sexually compatible. So here comes the limitation of this technology, though it has given us enough confidence, enough product, and surprisingly very good progress when it comes to ensuring the food production for Earth for the past 50 years, I would like to say specifically. And again, I would like to say that this is a technology which has solved Herculean tasks. Herculean tasks, because I'm going to show you some data, because based on this technology only, we could actually survive especially in India, when we were importing a lot of food and famine were there in our country. Not only that, in terms of food production, it has actually modified the plant genome thousands of years. The plant breeding has resulted in diversity, which we see in terms of color, in terms of the size of the kernel, in terms of shape of the fruit over here. If you see, maize is shown over here, carrot is shown over here, and cauliflower, purple color, is shown over here. This is all because we could achieve the target because of modified plant genome. Because when you are doing the pollen breeding, the classical plant breeding, you're actually creating new genomes by allowing genome from one plant to cross or mix up with the plant or genome of another origin. In a way, you're creating new genomes, and that is reflected in the form of different color, different shape, different qualities, different nutrition qualities as we are talking about in this process. To further strengthen what I'm talking about, I'm going to show you this example, which is a classical example all of us feel happy about. Shown over here is on the right side, if you see, where I have written the bread wheat. This is the modern day wheat variety, and I request that you should see the number of seeds per panicle, the length of the awn, and the total yield in these bread wheat. And if I tell you that the plant on the position 4 is actually a baby of plant shown in the position 3rd, and the plant on the position 3rd is actually a baby of position 2, and position 2 plant is actually a baby of the plant which is shown in position 1, which is shown on the extreme left side of it. And if I now ask you to compare the plant on the left side, leftmost side, to the plant on the rightmost side, most of you will not agree that this is an achievable target. And if I tell you, yes, this has been the case, this baby and the father and the forefather have undergone a lot of domestication, a lot of breeding experiments, and that has given, if you compare the number of seeds per panicle over here in one panicle, in case of monocot like wheat and rice, we call it the inflorescence of the panicle. The number of seeds per panicle, if you see the shape of the panicle, the length of the awn, which can be seen, and has been drastically changed by the breeding process. And what we have in front of is a modern wheat variety, which we call domesticated varieties. And this has given us more food per plant, more food per drop, more crop per drop. A lot of slogans are there already in the pipeline. But nonetheless, this shows the power of the technology. We could handle it, we could produce more because we understood the simple process of plant breeding, and we could achieve the higher yield in crop plant in years to come. Another example, which is again a classical example, I would like to show you. The modern corn is shown on the right side. The circle over here shows the cob, and the bigger circle, I'm not able to use the pointer somehow. The bigger circle on the right side shows the cob. And if you see very carefully, I have kept a rupee coin over here. And the rupee coin has been kept purposefully in the picture just to have a comparison, because rupee size on the left picture, on the right picture is same. But if you compare the size of the cob on the left side and the right side, the number of seed per cob, you will be amazed to see that yes, the crop, the cob which I'm showing on the left side is actually father of the baby, which is shown on the right side. Be it in terms of number of leaves, if you compare the plant architecture, which is there in the teosinte, the wild corn we are talking about, and by the process of breeding, this has been modified so drastically by crossing different genome that we have more productivity, good quality of grain, more grains per cob, and a lot of good characters they have been in there, giving us the confidence that yes, we can actually do a lot of wonders when it comes to handling biology as well, not in terms of physical engineering, as I showed you the removal of mountains and making of dam. We can actually do something in biology as well, which is giving us confidence that yes, we can handle the Herculean task of feeding 9 billion. I'm going to give you more example in subsequent slide to stress upon what I'm talking about. If I show you the watermelon which you took morning in the breakfast, the modern watermelon shown over here, and the traditional 9 and 1645 is shown on the left side. And I'm sure none of you would like to eat the one which is shown on the left side. But this is all because we dreamt of it, we took it as a challenge, and now we have the product available. I can continue showing such example for next 1 hour, but I'm just showing you some examples so that you get the confidence in the technique. This is the tomato which you every day use it in the house. Please pay attention to the wild rice, which is a small tomato which is shown on the right bottom of my slide. And if I tell you that the picture has been taken together, where the cultivated tomato is a huge one, at least 100 times bigger than the wild tomato. It's not that I'm not saying that these wild tomatoes are not important, because they're important when it comes to certain genes pool associated with the quality, nutrition, anthocyanin, but nonetheless, it has given us good product coming out of it by the process of classical plant breeding. Sorry. This is the diversity which we have created in many other crops, and shown over here is a chili. We have carrot on the left side, we have banana in the middle, and we have the eggplant on the right side, giving us confidence that yes, we can continue doing that. And here are some facts which tells us that in India, if you see, our performance has not been so bad. Rather, it is extraordinary. If you see the column which is showing the productivity in the year 1950, close to our independence, we could only produce food grains as good as 50 million tons. But now, if you see the productivity in the last year, 2021, 2022, you'll be surprised to know that we could actually produce more than 330 million tons of grains. Pulses, again, if you see, 3.2 times. Oilseed is 7.2 times. Cotton is another example, 11 times. Sugarcane, 7 times. Horticulture, 3 times. Milk, 11 times. Fish, 18 times. Egg, 63 times. Meat, 4 times, and so on and so forth. The reason I'm showing it you is that we could do it because we took the challenge and we had the tools and technologies available, and we could actually make our country, begging and bowling in front of other countries for the food when it was there in the situation in the 1950s. Now we are exporting the food to almost 16 different countries as on today. These are some of the other details: rice, wheat, maize, cereal, oilseed, sugar, cotton, jute. And if you see any of the comparison, we have not, uh, we have not actually uh, shows any downfall in our productivity. We are increasing. And remember, this is happening despite the fact that less and less land is available for agriculture because of urbanization. And in despite of that, our productivity is going very, very high. Then the question comes that if we have shown so much progress, then why are we worried about it? Why are we even talking about it that this is the problem? My dear friends, those who are young colleagues joining from different parts of the country, I'm going to pose you a question over here, a challenge over here, the problem over here for which your generation will have to find an answer. And the question is being shown in the form of two simple lines which are drawn in the graph. On the left side, you have a graph which is showing the global population. And if you see with time, it was not so big to the year 1950, 1940s, but suddenly then it woke up and it started rocketing, shooting high in the sky. This is the real challenge in front of us. This is bothering everyone. This is making us mad when it comes to handling so much population, urbanization, feeding, making houses, making medicine, and so on and so forth. And on top of that, what is shown on the right side is another graph which is talking about atmospheric CO2. And I'm not showing the global temperature over here, but these are the results of the human activity when we are worried about the global warming as well. Taken together, these two problems will have a synergistic effect, meaning that by that we need to produce more and more despite the challenges of high temperature, high CO2, with less and less land available because we have more mouth to be fed. So the question as a biologist you should ask yourself, and we are doing that as a scientist, as a plant biologist, that how do we feed more people without further damaging our planet? Because the global warming, I personally consider, the green revolution has been good, but the global warming, which is affecting, which is being predicted, or we call it climate change as well. These are thing which are actually damaging our planet. And this is a question I think we need to ask ourselves very carefully. I'm going to show you some fact over here again. There is a website, eri.org or World Bank, where you can get this data from. There is a clock which is ticking every second. This is an old figure. I'm sorry, I should have revised it. This is around 8.3 now, 8.3 billion. So the world population is 8.3 billion now. And if you look at the clock, which is a real-time clock, you sit in front of your monitor, and this clock goes like tick, 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 tick. It is very scary to see that we are increasing in number by such a speed as far as global population is concerned. Another concern is also related to the kind of food habits we have. And if I tell you that almost half of these people, roughly 4 billion people, depend on rice as a staple food. And this has a lot of implication in terms of crop productivity, in terms of monopoly. But what is more important for us to understand and ask this during this meeting, this discussion, is that there is another clock which talks about productive land in hectares. But unfortunately, this clock is not growing. This clock is not ticking in an increasing manner. We can't even increase the green land on our planet Earth by even 1 millimeter or 1 micrometer. The land available is limited, and on the other hand, the land which is available for agriculture in terms of productive land is decreasing. This clock also goes like tick, 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 but it is a decreasing in number, meaning thereby that, as I said, we have to produce more and more from less and less resources, less and less productive land available. if we really want to sustain. This is the Herculean task I'm talking about. Today we have enough food available. Our FCI godowns are full of food. But think of a situation. It is being estimated that by the year 2040, India would need at least 70% more food. And already we are the number one country in terms of population. We have surpassed the China in that. And so coming to our own country, situation is very, very, very difficult. And on top of that, when it comes to agriculture, there are environmental abuses, which we call stresses, be it in terms of drought, which was seen in some parts of the Karnataka and other places last year. I was told that 34 districts were declared drought prone, and states such as Punjab were actually suffering the flooding during the last monsoon. So on one hand, we have unpredictable rain, but the water which is also being used for agriculture is turning saline. So high temperature, low temperature, salinity, drought, heavy metal, viruses, insect, nematode, a lot of things are actually enemy of the crop productivity, which we call stress, and they result in decline by almost 70%, meaning that by that, if the crop potential is to produce 10 tons per hectare, but the farmer is actually able to harvest only 3 to 4 tons per hectare, the 60 to 70% decline, or the yield gap, which we call it, is there between the potential yield and the actual yield. And that makes me worry as a student of biology, the student of plant biology, that can we ensure food security for our next two generations? That's the question we need to ask. And this is where my title of my presentation, Are We Ready to Feed 9 Billion, is justified. Now, since the title is justified now, I'm going to show you some of the success stories where we have adopted new tools and technologies to come up with a new solution. The solution so far I showed you is by using one technique, which is breeding, breeding, breeding. But as I said, the breeding technology is actually suffering from limitation because you cannot cross one rice plant with any rice plant, or you cannot cross mango with rice, or carrot with wheat on something and something, right? So every technology has a limitation, and we cannot rely on one technology as far as challenging such Herculean task is concerned. That is the reason we need to have more and more solutions coming up, more and more technologies coming up in our hand. So this graph is something which I want to share with you, which talks about what I've already mentioned, that we will need at least more food. And shown over here is a statement which was made by Clive James. He visited Jawaharlal Nehru University in the year 2016, and he made a statement based on the fact that in the next 50 years, mankind will consume as much food as we have consumed since the beginning of agriculture 10,000 years ago. Believe it, read the statement again if you have not read it. It talks about a very, very hard fact, very important hard fact, very alarming hard fact. In the next 50 years, mankind will consume as much food as we have consumed since the beginning of agriculture 10,000 years ago. Where is it going to come from? Who's going to produce that? And that is the question which bothers me at the moment. When it comes to countries such as India, we are very special. Of course, we are special, and we are special in many ways. One of the ways that the population of many of the states, which is listed on the left side of the first figure, Uttar Pradesh is equivalent to the population of Brazil. Population of Maharashtra is equivalent to population of Mexico, and so on and so forth. Meaning thereby that we are not feeding 27 states in our country. We are actually feeding 27 countries within our country. Can you believe it? We are feeding 27 countries within our country. You would have not realized it, this hard fact, but yes, that is true. And not only that, most of us do not have access to nutrition food. In fact, many of us do not even understand the meaning of nutrition food because we are typically depending on rice and wheat for calories. We do not have access to fruits. We do not have access to pulses. We do not have access to the vegetable. And this is what our India is. That means that India is facing a big challenge as far as food and nutritional security is concerned. And in years to come, we have to have new technologies which are going to give us not only food security, but nutritional security as well. And on top of that, there is a shift in understanding that the number of farmers are more interested in adopting other challenges. They are shifting to other professions, and they are more moving towards labor intensive farming, not doing the farming themselves. And if I ask you to close your eyes and imagine a farmer in our country, not only in India, but many of the southwestern countries, South Asian countries, you will be actually imagining a farmer with, not sure about the mountain, praying to some superpower, expecting a good mountain this year, and return of investment where he has made investment in terms of buying seeds and other things is not guaranteed, and most of the time you're totally dependent on the productivity and that dependent on the monsoon. And this is where I think the limitations are. And some of these images are very common in many metropolitan cities. It's very disheartening to see this kind of situation which is there in our country at the moment. But we need to, ensure that these people are able to get good education, good food, and this is where I think we need to prepare ourselves in years to come. When it comes to achievements, I think I don't have to tell more about the Green Revolution, the efforts of M.S. Swaminathan and Norman Borlaug. M.S. Swaminathan passed away 2 months ago at the age of 99. They were actually responsible for saving India in the time when we had no food available. We were running through famine, and they brought in the Mexican varieties, crossed them with Indian varieties, produced the dwarf varieties with higher yield, and that's what we call green revolution. That's the first development as far as agricultural biotechnology in India is concerned. Second development is in the form of hybrid seed, when we learn that there is always a hybrid vigor when you cross two distant varieties. Third one is the biotech crop, when we learn that you can go beyond the classical method of breeding. You can actually insert the gene from one crop to another plant, provided the gene is known to be safe and its function is known to that. So now, next part of the talk, I'm going to show you in next 10 minutes. I'm going to show you some success stories as far as agri-biotech in India is concerned, using the modern genetic modification, which is simply defined as inserting one or few genes to achieve the desired trait. I'm using the word one or few gene because some of the traits are controlled by more than one gene. And this is how you insert a gene, by using the bacteria, classical bacteria, agrobacterium, polyethylene glycol, microinjection, electroporation, particle gun. Those of you who had a chance to look at these kind of technique or have practiced those things, they will realize that yes, we have done a lot of advancement when it comes to developing transgenic plant. And the method for gene transfer have also evolved in due course of time. But nonetheless, the bacteria mediated, agrobacterium mediated transformation, I will not go into detail of these, which I wanted to because of time paucity. But nonetheless, there are methods available which can actually be used for gene transfer in plant. And besides that, you have other techniques also for crop modification. Crossbreeding, I just mentioned it to you. Mutagenesis, where you use ethyl methyl sulfonate or radiations to create random mutation. Those mutants are then screened and then used as a variety. Polyploidy, where you add colchicine, where the chromosome number double and the fruit size increase. Watermelon is a good example. Protoplast fusion, where you can actually inherit the maternal trait because they are controlled by the chloroplast and mitochondria. So protoplast fusion has also been attempted. Transgenesis is a technique which allows addition of genes from any species to anyone to create a new variety of desired trait. And the most recent one is the genome editing. And I'm very happy to say that Indian government has created a very enabling environment where the product coming out of genome editing, where you do not leave any piece of foreign DNA, and you use an enzyme which can even modify a single base pair directly within the cell. The government of India is proactive in allowing such kind of interventions and giving space to such innovation. And they have permitted the product which is coming out of genome editing, especially the SDN1 and SDN2, where no foreign DNA is left, and they have considered that this is as good as the conventional breeding. So this is a very bold step the Indian government has taken, and we are very thankful to government for being proactive in that. Nonetheless, these crops are being utilized in almost all countries, and I'm going to show you some examples. The first example, I think all of you are aware, is the BT cotton. This has become a newspaper item. Government of India introduced 2001, 2002 the BT cotton, and since then the productivity, if you see, it's going higher and higher, and we are the largest exporter of the fine quality cotton for the rest of the world. This technology is actually again driven by a form of a phenomena of we call it bio pesticide. the gene comes from a bacteria which is toxic to the insect, Lepidopteran insect. Shown over here on the left side is the insect which causes havoc, especially in many crop in cotton. And if you see the cotton which is eaten by the insect, which is shown on the right side, um, this infestation is really devastating. Not even a single fiber is left, and the farmer, they are not able to even make a return of investment. So there is a gene which is coming out from the bacteria which we call cry gene. This codes for a protein which we call cry protein. So what scientists did, they isolated this gene from the bacteria Bacillus thuringiensis. That is the reason it is known as Bt gene as well, and introduced this gene into the crop plant, where it started producing the protein corresponding to cry gene. So instead of now bacteria which we used to spray on the plant to kill the insect, we are now plant producing the protein themselves, which is killing the insect which are coming and eating them. So this is very interesting to know that. If you see on the left side, you have the corn hybrid with a Bt gene and a hybrid susceptible to European crown borer. On the shown on the left side, the figure I'm talking about is shown on the left side. Within the figure, left and right, I'm talking about, and you can see the difference between the plant on the left side, corn plant, and plant on the right side. And the cotton, if you see, in the one ball on the left side is non-Bt, and on the right side you have a Bt, and you can see that there is a hole in the Bt cotton as well, which means that insect came, it started eating the plant, but when it started eating the plant, it died. So contrary to what most of the people believe, that Bt plant will not allow the Bt plant will not allow the insect to come. But yes, you cannot stop the insect to come. They will come, they will eat, but once they eat, they will die because of the technology. And this technology is quite safe for human being because the protein is active only at the pH when which is highly basic. You and I, as a human being, has very highly acidic pH, because as you can see, when you eat a lot of chatpata food or you take more tea, acid comes out of it, because so it's because we have acid in our stomach. But insects, since they do not have acid, they have basic pH, so this protein is only toxic to the insect because of the pH activity. And I don't have to tell you that pH is very important for any enzyme to be active at all. This is transgenic virus resistant papaya. This is Roundup Ready soybean, herbicide resistant soybean. This is salt proof tomato. The traditional tomatoes are shown on the left side, and the salt proof tomato, which are able to sequester high amount of sodium, is shown on the right side. Otherwise, the plant do not survive under salt condition. I do not have time to go into detail, but any of you, if you would like to know more about it, I will leave my email address with you, and you can talk to me later on. This is an interesting story of golden rice, which is not only golden in color but golden in qualities as well, because it has higher content, very high content of provitamin A. And vitamin A is very important when it comes to eye health, and it saves the life of many people. And this has been the reality now. Uh, Tomato varieties with enhanced lycopene, which is a kind of vitamin A and a very important antioxidant, is also there. The tomatoes, when they are brought to the home, they ripen very fast. So delayed ripening has been introduced in tomato because, for the purpose of having a enhanced fruit life. Oil quality and content have been improved by having vitamin E into them. Plant-based vaccine are going to be reality, because the antigen is being produced by the plant, such as banana or the watermelon, and they gives us the confidence for diseases such as cholera, hepatitis B, diarrhea, and investigations are going on at the moment. Decaffeinated coffee is a reality now. It is available in many of the state, many of the countries now, which do not have caffeine. We have good quality of grapes, which is coming up. Nicotine-free tobacco is a reality. Blue rose is a reality. Gluten-free rice is a reality, and so on and so forth. I can continue giving good example. And these were not possible by the classical breeding, but the technique of genetic modification allowed us to have these kind of products available. So what I'm trying to say over here, that yes, we could achieve the production of rice and wheat when we had the green revolution by using the technique of classical breeding. But now, since the challenges are new, so we have to adopt new techniques. And here is a technique which allows us to be supporting the classical breeding and giving a solution, making us less worrisome for the future when we talk about feeding 9 billions. The modern plant sciences is no more a science of plant breeding, but it is a science of analytical instrument, high-end analytical instrument, such as GCMS, HPLC, and so on and so forth. And highly skilled manpower is required to analyze the sample and to come up with some tangible information, because it requires a lot of use of algorithm and bioinformatics tool, computational tools. So no more classical plant breeding is the only way, but we are talking about modern plant biology where we have DNA, we have protein, we have metabolite, and we can go deep into a cell to ask a question and find a solution to a problem. That means that we have more confidence as on today in terms of making products which are good for human being consumption. Those who are from the field, they'll be noting down some of these names of advanced genetic technology which have contributed to improved yield. I will not again go into detail, but these tools such as marker-assisted selection, genome-wide association analysis, recombinant DNA technology, cisgenic, transgenic, transgrafting, precision genome editing. These are the techniques, tools giving us confidence, which I just showed in the beginning of my presentation, which are going to be as useful as the simple mining process could be achieved, or simple making the dam largest dam is concerned, because these tools are going to give a solution when it comes to having food available for masses in years to come. Another technology I would like to mention pertains to genome sequencing. India participated in RISE genome sequencing. University of Delhi South Campus and IARI sequenced the chromosome number 11 in International RISE Functional Genomics Consortium. But nonetheless, several genomes have been sequenced, including the ones which are native to our country, such as mango, such as neem, and neem, which we use for diabetic people, the maize, rice, the soy, brassica, grapes, you name any plant, and it has been sequenced. What I mean by the sequence, for those who are not able to understand the meaning of sequencing, we are trying to decode what is there in this plant in terms of the DNA sequences. We know that DNA is composed of four bases: adenine, guanine, cytosine, thymine, but the sequence in which these nucleotides are arranged determines whether a plant is going to give higher yield, or a plant is going to be insect resistant, or a plant is going to give me higher yield. A lot of things can be understood by digging out the genomic information, as we are doing it for human genome as well. Yes, the modern technology, plant breeding, is no more restricted to putting pollen onto the stigma. It requires a lot of understanding of phenotyping, genotyping, association analysis, and use of high-end computational tool to have gene discovery coming up. And those who are more familiar, more happy to see the modern tools and technology, I'm here to say that the drone technology has a lot of application in modern agriculture as well, because the cameras attached to these drone cameras, be it infrared cameras or hyperspectral imaging, they can detect the diseases right at the point of inception. And as soon as you detect a particular plant is a disease by taking aerial pictures, you can uproot that plant and burn it out, so that your virus or the bacteria do not spread to other plants as well. And you can go beyond this by predicting the level of water in the soil, the level of pH in the soil. A lot of things you can do by using different kind of images. And AI and computational biology are finding, big data analysis is finding new roles in agriculture than any other field besides health sciences. The technique of gene editing, where you are able to use enzymes such as CRISPR, Cas, talons, zinc finger, they are making revolution as far as going deep into the cell, into the nucleus, into the chromosome, and cutting out, as I'm showing with a scissor, particular base pair and replacing it with a new one, so that it's kind of, like it's kind of a gene therapy. If somebody is having, say, some disease, you can take out that bad gene and put a normal gene or a good gene into that. And that is what we are doing it when it comes to genome editing, without leaving any foreign DNA into that. So these kind of revolutionary technology are giving us a lot of confidence that the future is very, very bright. And the credit for these discovery goes to these beautiful ladies, Doudna and Charpentier, who won the Nobel Prize in the year in chemistry in 2020. And this technique has actually revolutionized the way we look at agriculture as on today. I will not go to detail of it because of shortage of time. But I'm going to show you this memorandum, which was taken out by the government of India in March 2022, where they said that the government of India has exempted the genome edited plant falling in the category of SDN1 and SDN2 from the provision of Rules 1989. Meaning that by that, these products which are coming out of SDN1 and SDN2, they do not have to be approved by RCGM and GAC. They can be approved at the institution level. And this has actually given a new breathe to the industry, agri industry, and many other countries in our neighboring countries, even Europe, which was so against genetic modification. After this verdict which was given by government of India, they have eased out, and they have also given permission for SDN1 and SDN2 in there. And some of the products which are coming out from gene edited are very nice. GABA enriched tomato. GABA is an antioxidant and very good peptide for human health. GABA enriched tomatoes are reality. Butterflies with a lot of new traits are a reality. Fish is a reality. Chicken is a reality. Goat is a reality. So you think of any problem, any disease, the only thing requirement is that you need to find out which gene actually has to be replaced. So like you and I have thousands of genes. Rice has 25,000 genes. The question which is still challenging at the moment is that which gene to engineer. So rather than focusing on the technology to change that gene, we are focusing on asking a question, which gene to engineer, which is the most suitable gene to answer this question, or ultimately which gene will give me the desired product. So that kind of question we are asking. We have enough tools and technology though available with us. It has a lot of application in agriculture, be it in terms of increasing crop productivity, resistance to diseases, improving product quality, abiotic stress tolerance, resource use efficiency, better nutrition. A lot of things can be done, and as we are moving through, sailing through, we are realizing more and more potential of this technology. Are we ready to harvest benefit in India? Yes, in India, we have a lot of laboratories which are working, a lot of product, a lot of crops are being generated. I will skip the details of that. This is not required for this forum, but yes, yeah. So conclusion is that the CRISPR-Cas9 genome editing offers a great potential. It is a step ahead, and we are very happy that the products are going to be available in India coming out of SDN1 and SDN2 categories. So starting from classical plant breeding, which I just showed you, a male and a female partner, we have come to a stage when we have more confidence in handling the problems related to multigenic trait, the problems related to diseases, the problem related to yield, and so on and so forth. So the message to the young generation, I'm sure many of you will be starting your research career as well. The future is here, right? The time is quite ripe to handle the problem. The time is quite ripe to answer these questions because we have the tools and technologies available with us. And it is believed that since we need to be 5 trillion economy, out of this, actually, agriculture has to contribute 1 trillion. And if we need to contribute 1 trillion, we have to have the right questions being asked, right technologies being used, and so on and so forth. Yes, I will skip the detail because of time limitation. In the next 10 minutes or so, I will just tell you what we have been doing in our laboratory at JNU, Jawaharlal Nehru University. It will be only 3-4 slides. As I said, abiotic stresses are extremely, extremely important, which includes high and low temperature, drought, salinity, heavy metal, and so on and so forth. The question which I'm asking with my laboratory is how to tackle the climate change problem, because the soils are becoming more and more thirsty, the water tables are going down, and whatever water is available, it is not good for agriculture. What to talk of being using for human purpose. So the question is that we need to make use of the land, which are fallow land, which have saline water, where saline soil is there. The soil quality is not good. The pH is not good. It has a lot of salt accumulated. So with this theme, which is total number of students we gathered in one of the function, in one of the day at our laboratory, we actually asking a question for the last 2 decades that how can we produce plant such as wheat, such as rice or brassica under conditions which are not suitable for agriculture. I mean that we are trying to make climate a resilient crop, and this is where we see that salinity and drought are major issues. This story was published on the cover page of scientific journal Nature, where if you see the circle, which I'm showing over here, it constitute the regions such as Haryana, Punjab, Rajasthan, and UP. The water table has actually gone down. I do not know how many of you have heard that Dr. G.S. Khush, who is a internationally known plant breeder. He visited Ludhiana last month, 2 months ago, where we have made a GS Kush museum as well. He said that if the farmers in Punjab continues to dig out water or harvest water at the speed which we are doing, within 10 years, Punjab will be left with no water even to drink. Punjab will be left with no water even for drinking in 10 years from now. This is what he has said. And this statement is quite alarming. Not only the water table is going down, but whatever water is available, it is not good for agriculture. It is turning saline and the pH issues are there. So in my laboratory, we are following two kind of approaches. On one hand, we are actually using the tools of reverse genetics, where we manipulate single gene or double gene, and we have battery of genes, almost 2 dozen patents we have got. The name of the genes are shown on the right side. And on the other hand, we are also doing forward genetics, where we are doing mutation breeding to achieve the same target. And we are working on rice, because rice, as I mentioned in one of my slides, it is food for half of the population on this globe. And I have no hesitation in saying that it is the food for richest and it is food for the poorest. So we are exploring the diversity which is available in our country, and I'm happy to share that International Rice Research Institute, which is there in Manila, they have more than 1,65,000 of rice varieties, and half of them have been donated from India. The rice is very diverse in India. It is being grown from Kashmir to Kanyakumari and from left to right in our own country. So you see a lot of diversity, be it in terms of not only the color, the size, and the shape, but in qualities as well. And this is the diversity which we have exploited. We have used a traditional rice which is grown in seawater, because we wanted to make the plant salt tolerant. So we realized that the farmers in our country in coastal areas such as Kerala, they have been growing a traditional rice variety which is known as Pokali. We almost studied this particular plant for 10 years, isolated some of the gene, developed the technology, and I'm very happy to say that this work has of the work the student have been this work of the student have been recognized in many awards at national and international level, be it in terms of Reliance Platinum Jubilee Award. And I'm very happy to say that a couple of years ago, in the year 2018, we were invited to Rashtrapati Bhavan to receive this technology award for development of a variety of rice which we call strong, STRONG, which stand for stress tolerant rice of the next generation. This rice gives you 30% higher productivity under saline and drought condition. This is really useful, and this technology has been transferred to an industry, and industry is very happy with it, and we are happy to share that yes, this technology is going to be reality very soon in years to come. So with this, I will stop sharing the slide and take questions for the next 10-15 minutes, if I can do that.
Participants, please ask question.
Good afternoon, sir. Good afternoon,
Very
sir.
good.
I am Kamachikala from Parasakti College, Kuchalam, Tamil Nadu. So, sir, it's really wonderful session. Thank you for your presentation, sir. You talked about many modern techniques to improve the crop production and nutritional quality of the crops. But I have one doubt about the quality of seed, quality, vigor, and germination of the seeds produced by the modified crops.
So what is the question? I didn't get it.
Sir, I want to know about the quality of the seed, quality, vigor, and germination of the seed produced by the modified crops.
No, what do you want to know? I mean, what is the question? You're telling you want to know about the seed vigor, quality.
Quality and germination of
Sir?
seeds. Yes, sir.
So are you doubting the quality in seed vigor?
No,
Is that
sir. No, I
the
don't.
question is
I
not
don't.
clear to me. What do you want to know?
I want to know about the germination of the seeds, quality, germination of the seeds
Germination of
produced.
seed is dependent on the lot. I mean, if it is an old lot, it will not germinate. If it is a newly harvested crop, it will not germinate, because every crop has a dormancy and life cycle. So with age, the dormancy with age, the germination will go on decreasing. And if it is a fresh harvest, it will not germinate. The dormancy period is also to be taken care. But yes, I think what you are talking about, that modern varieties, they have some reservation or they have some implication in terms of germination. No, I don't think so. Nobody has reported that.
Okay, sir. Fine, sir. Thank you.
We have been doing classical breeding only. I mean, in terms of understanding these plant behavior, we are now doing it through genes only. That's the only difference. But so far, nobody has reported any different in terms of germination, in terms of quality, because you are only targeting a particular trait. You're not targeting a trait which is related to quality, or unless you have specific gene which is involved in germination or involved in the nutrition quality. But otherwise, I think there's no difference between the traditional varieties and these modern varieties. That's my submission to you. Maybe all of you can turn on the camera so that it's a good. Those who can turn, I mean, some of you at least can turn camera so that I'm talking to human being. Otherwise, it looks like I'm sitting in front of a screen and not in front of any human being I'm talking to. Yeah, some of you have turned on your camera. Great. Any more question before we call it a day?
Sir, good
Yes, please.
afternoon.
Good
This is
afternoon.
Dr. Siddhi Jalmi from Goa University, and it's honor to listen to you, sir. Although I have heard you a lot of time, because I was in NIPGR doing PhD under Dr. Alok Krishna Sinha. So
Oh,
during
okay.
that time, I've listened to you. So we are, as a molecular biologist and biotechnologist, we are constantly talking and promoting the use of
Hello.
RDT and genetically modified crops. But how
Acha.
easily they are accepted till now? Like,
So that's not the job of scientists, I would like to say. The scientists, our job is to keep on creating that. Let's leave it to the social scientists and policy makers. Of course, the government of India is changing environment, and a lot of new things are coming. You already know about the mustard, which is being developed by Dr. Deepak Paintal, the GM mustard. So government is totally supporting. As the scientists, we are sure of the technology. What happens after that is none of the science, and we should not worry about it.
yes, sir. Thank you, sir.
Acceptance is a challenge at the moment, but I think time is going to change the perception. Today we have a choice, tomorrow we may not have choice. Like it happened with the COVID vaccine. None of us would agree to, get a shot of a vaccine which is develop a shot at such a short notice without different level of trial they have done. The RNA vaccine is another good story, but uh, we had to do it because we had no other option. So COVID vaccination is a good example. Our thinking changes when there is emergency. So let this emergency not come with crop productivity, I would like to say.
Yes, sir. Hoping for
Let's
this.
not wait for the
Yeah.
famine to come to be ready with the product.
Thank you, sir. Thank you so much.
No doubt. Good question.
So, what about the mutation rates, like after the gene modifications and stuff? Like the
Your voice was not clear.
so the possibilities of what are the possibilities of gene mutations after certain times or in this modification?
Gene mutation, they're as good as any traditional rice. I mean, why would you expect that gene mutation rate will be different in these varieties? I mean, when it comes to evolution, all the genomes are evolving. Be it a product developed by classical breeding or genetic modification, be it human, microbes, evolution is part of succession. So evolution would happen definitely. But I don't see any reason to worry about mutation. It's not that the plant is not going to mutate on its own unless we treat it with some mutagen.
Okay, sir.
They're not different from the traditional.
Okay, sir. Okay.
Yeah, I suggest that any of you have any question, please clear the doubt. You do not keep the questions with you. Otherwise,